Perfect, thank you. So it's a real pleasure today to introduce to you Professor Sir David Klenerman. So Professor Klenerman is a researcher here in Cambridge, based at the UK Dementia Research Institute, and his lab focuses on applying biophysical methods to biomedical problems. So this is something that's very relevant for TMS. So our group, just to explain to you as well, also is an interdisciplinary group. So we're primarily biologists and biochemists, but we also have a real interest also in physics and computer sciences. So your research really brings all of that together today, which is really nice. So Professor Klenerman is rather well known in the field of sequencing, as he co-founded and invented Selecta sequencing, which is now more commonly known as Illumina dye sequencing, and has also been very heavily involved in developing methods to image individual proteins in living cells, which I think he's going to tell us a bit more about today. So it's a real pleasure to have you here with us for TMS. And without further ado, I'll pass over to you on to begin your presentation.
Thank you very much. So hopefully you can all see my screen. So I'm going to talk to you about how to image and characterize protein aggregates associated with neurodegenerative disease. So this is an image of live T cell receptors diffusing around on the basal surface of a T cell. And later on in the talk, I'm going to show you how we get these images. So the outline of the talk is I'm going to introduce you to single molecule fluorescence and super resolution microscopy, and explain how we use these methods. And then I'm going to show you how we can develop and apply these methods to study protein aggregation and neurodegeneration. And the main message, I think, of the talk is how new physical methods enable new science. So if you can detect things or image things that no one else can see, you hopefully can get new insights into your biological problem. So just to make sure we're on the same page, I'm sure you all know this, there's a fundamental limit in terms of what we can detect using light. This is the so-called diffraction limit, which is about 0.2 microns. So this is just a length scale, and things below this diffraction limit are things like viruses, protein aggregates, or small molecules. And one of the things I'm going to show you is how we can image things that are smaller than the diffraction limit and still measure their size. So what we do, as shown in this speeded up video, is build our own instrumentation because you can't buy it. So this is Alex and Anna building one of our microscopes. So our microscopes are based on an inverted, hopefully you can see my mouse, an inverted microscope here, a whole bunch of optics here and lasers. And so our labs are very colorful. We have a bank of lasers, purple, blue, green, and red, which we shoot over using optics to our microscope in order to do our experiments. So in these experiments, we have light that undergoes total internal reflection at a cover slip, and this produces an evanescent wave that penetrates about 200 nanometers above the surface and enables us to image molecules 200 nanometers, within 200 nanometers of the cover slip. So these are molecules diffusing around on the cell surface. And this is how we obtain this video of the T cell receptor that we, in this case, labeled with a FAB, which had a dye on, diffusing around on the surface of a live T cell. The other instrument that we built is based on a light sheet. So the light comes in from the side and we produce a thin sheet of light. And above the cell, we have this nanopipette, a very fine glass pipette with an aperture of about 100 nanometers. And we can use this to deliver protein aggregates to the cell surface and then follow how the cell responds to these aggregates. We're particularly interested in understanding how these aggregates induce an inflammatory response. So just to show you we're good at delivering molecules, this is a video where we're delivering fluorescently labeled wheat germ agglutinin onto a glass surface. And we can do this under computer control. This is why we build these instruments, and we can make pretty patterns. Oops. So what we can do using this is, sorry, I just want to check one thing. Sorry about this.
No problem.
Right, seem to be using the wrong slides. So what we can do is with this is on the surface of a cell, it's this toll-like receptor, and this binds to LPS, and it binds to protein aggregates, and it forms a so-called midosome, which is a signaling complex. And in these cells, we've attached GFP to the MID88. And when we form these midosomes, if I just pause this, so we use the pipette, we deliver the aggregates, and we see formation of these midosomes. We see the GFP of the MID88 come together to form this signaling complex in real time on the surface of the cell. So you can see these midosomes build up on the cell surface. And once the MID88 has formed a midosome, there's a downstream signaling that ends up with NF-kappa B translocating to the nucleus and production of pro-inflammatory cytokines. And we think, as I'll show you later on in this talk, that the triggering of toll-like receptor 4 by protein aggregates plays a key role in Alzheimer's disease. So I'm now going to show you how we perform super-resolution imaging. So this is a normal diffraction-limited image of a sample. But if you organize things that you only image sparse molecules, they're still blurred by diffraction. So they still show up as a 200 nanometer diameter spot, but we know it's from a single molecule. So we know the molecule must be at the center of the spot. So we can replace these blurry spots with these points here where we've shown where the molecule is. And we can do this with much better precision. Typically, we can do this with 20 nanometer precision. Then we repeat the experiment again and again, and we can build up a super resolution image of the sample. And this has been one of the big revolutions in the field of single molecule fluorescence, this ability to image things at super resolution. So what I'm going to show you here is the T cell. This is what it looks in the white light image. We fixed it, and we have weak germ agglutinin that binds to the cell surface. And we're doing this in 3 dimensions so we can image the topography of the cell. So we take slices through the cell. And then we can reconstruct the whole cell surface. And hopefully you can see that there are these fine protrusions sticking out of the cell surface, so-called microvilli, so that the T cell uses these to sample the antigen-presenting cell. So this looks very much like what you see in electron micrographs of T cells. So what we can do now is image the T cell receptor on the cell surface, and we can image the cell topography again, and we can ask, are the T cell receptor randomly distributed over the cell surface, or are they particularly clustered at the tips of these microvilli? So the red spots are the T cell receptor, the green is the cell surface, and again we can build up super resolution image now with the T cell receptor mapped onto the topography of the T cell. This is all at about 20 nanometer resolution of a fixed cell. And we can then ask, are they randomly distributed and localized in the microvilli? And they turn out to be randomly distributed. So I'm now going to focus the rest of the talk on neurodegenerative disease. And the problem we're really interested in are which are the bad aggregates that cause Alzheimer's disease and Parkinson's disease. So this is not a cartoon. This is actually based on data that we've taken over the last few years, which shows that we have monomer. The monomer can come together to form dimers and trimers and bigger aggregates. And then these aggregates can undergo a structural conversion from these globular aggregates to these more fibrillar-like aggregates that we showed are much more toxic to cells and then grow on in order to form fibrils. And what we showed in one of our papers is this structural conversion is actually quite slow. It takes about 24 hours to switch from these globular aggregates to these more toxic proteinase K resistant fibrillar-like aggregates. But which of them are toxic? It's very heterogeneous. We have a whole range of aggregates of different sizes and structures, and we want to identify which ones are toxic and by what mechanism in order to know which aggregates to target in therapies and what pathways we should target for therapies. But the challenge is it's the aggregates at low concentration and abundance, and like I've just said, they're very heterogeneous both in size and structure. And we have a concentration gap. Lots of the studies, particularly in vitro studies, are done at much higher concentrations than occur physiologically. And so species are going to be formed that aren't going to be formed under physiological conditions. There's a reality gap. We tend to use a pure protein, where in reality the protein will be post-translationally modified and might also interact with other proteins present in vivo. And there's a time gap. Typically, we do experiments in a day, and the disease takes decades to develop. So we need to basically try and address some of these gaps. So what we've found is that we can actually detect the real aggregates that form in humans. And this was an experiment that was done by one of my students a while ago. So what Matthew did is we took a dye called thioflavin T, which is non-planar in solution, but it can become planar and highly fluorescent when it binds beta sheet containing oligomers. And what Matthew did is take some cerebral spinal fluid from a patient who was unfortunate enough to have Parkinson's disease, put it on our cover slide, and then added thioflavin T and imaged the thioflavin T active species using one of our sensitive microscopes. And what we found is these dots, these bright spots on the surface, and we found that there were more of these for the Parkinson's disease patients than the controls. And this opened up a lot of questions for us. What were these aggregates? Were they alpha-synuclein? What was their structure? And we realized that we could study the real aggregates that form in humans. We didn't need to use animal models. So our approach now is basically to exploit these to develop quantitative methods to detect and characterize individual unlabeled aggregates to elucidate the molecular mechanism of neurodegenerative disease. We can measure the number, size, structure, toxicity, how easy it is to degrade these aggregates, and their seeding ability. And the other thing that we do is develop models of the aggregation process in humans that we can extrapolate to long periods of time. So this is some examples of what we can do. Here we have aggregates on the surface. You can see these spots of different intensity. We add proteinase K, and you can see that the intensity goes down as they're degraded by the proteinase K. Down here, again, we have aggregates on the surface. We can add monomer, and these aggregates will grow, and you can see the spots increase in intensity as they grow. In this video here, we have prion protein, murine prion protein. And this is a speeded up video, but hopefully you can see the aggregate, it grows, and then you can see it break into two aggregates, which is a process that's supposed to take place when prions replicate. So you can see the aggregate here, and hopefully you can see it splits in two. So we're directly seeing this aggregate split into two aggregates. So this is an example of us super resolving a protein called tau, which binds to microtubules. This is the diffraction limited image, and hopefully you can see we get much more details when we do the super resolution image. So what we can do is study a process called seeding in cells. So the idea is you add tau fibrils, they're taken up by these hex cells that contain P3-1S labeled P3-1S tau, which is labeled with a divenous. And if we have the tau fibrils, they enter the cell, they grow, and fragment to produce a large number of fibrils. So if you look at this video here, instead of having all the tau and the microtubules, you end up with these fibrils formed all over the cell, somewhat a small number of which are diffusing around inside the cell. And we can super resolve these. So this is the diffraction limited image. And here you can see all the tau aggregates that form after we've performed the seeding. So you can see all these little fibrils that form. This is just a zoom, just to convince you we can do super resolution imaging. So that's a micron scale bar. So hopefully you can appreciate that we're seeing these very small aggregates inside the fixed cell. And here's another example. So what we can do is image before we add the seeds, we see very little, and then we can follow the seeding in the cell by imaging. And then we have a small number of aggregates after 7.5 hours, and a large number of aggregates after 24 hours. So the rest of the talk, I'm going to really focus on examples where we've applied this to, I think, quite challenging problems, cerebral spinal fluid and human brain samples, hopefully to convince you that we can apply these methods to real human samples. So the first example, we took cerebral spinal fluid from 10 control patients who were 70 years old in Gothenburg, 10 patients who were mild cognitively impaired, which is the early stages of Alzheimer's disease, and 10 patients who had Alzheimer's disease. And we used 2 assays that we developed to measure the toxicity of the aggregates. The first assay, we have a large number of liposomes arranged over a surface, and the aggregates, if they're present, can permeabilize the membrane. Calcium can enter the liposome, and it binds to a dye that's present in the liposome, binds the calcium and becomes fluorescent. So by measuring the extent of fluorescence, we can measure the relative number of aggregates that are capable of permeabilizing the membrane. So if we do that experiment, we get this low level with the control, this much higher level with the mildly cognitively impaired patients, and a level similar to the controls for the Alzheimer's disease patients. We did another assay where we take cerebral spinal fluid and add it to a microglial cell line and measure the amount of TNF alpha that these cells produce over time. In this experiment, the control and the MCI patients gave this low level of TNF, whilst the Alzheimer's disease patients gave this much higher level of TNF. So this seems to suggest that the mechanism of toxicity is changing. We get higher membrane permeabilization early on in the disease, and later on in the disease, we get a lot more inflammation. And we think that these changes are due to the aggregates that are present in the CSF. So if we take an antibody that binds A beta, what we found is that the N-terminally binding antibody reduced the amount of TNF, but not the C-terminally binding antibody, which suggests it's a fibrillar form of the aggregate, because the C-terminal antibody is not accessible in fibrils. But in contrast, if we add this to the calcium influx, we get similar reductions with the N and C-terminally binding antibody, suggesting it's A beta aggregates with a different structure which are causing the membrane permeabilization. If we do this experiment and then add an antagonist, either RSLA or TAC242, which binds to a receptor called toll-like receptor 4, you can see we can reduce the amount of TNF that's produced. So this all shows that the signaling, the inflammation is induced by fibrillar aggregates and due to signaling by toll-like receptor 4. In order to image the aggregate, we used an aptamer of DNA. So these are short 20 to 100 base pairs of DNA strands that are evolved using a select process to bind a particular substrate, in this case, fibrillar A beta or synuclein. So in these experiments, we bring the aggregates to a surface, add the aptamer, which we've extended with a docking strand here. So we put in the aptamer, and then we add a fluorescently labeled imaging strand, and this will bind to the docking strand, and then we can get see a spot in our image, which we can super resolve. Then it will unbind and rebind, so we can build up these super resolution images of the aggregates. This is a diffraction limited image of a small aggregate. This is a super resolution image. This is a fibril of alpha-synuclein, diffraction limited and super resolution. And again, this is for A beta. So hopefully you can see we can get super resolution images of these aggregates. So we did this for CSF. What we found is the number of aggregates was the same for the control, MCI and Alzheimer's disease patients, but the size distribution of the aggregates was different. So this is a cumulative frequency plot. So what this shows is that at this point here, half the aggregates are less than say 50 nanometers in length. And so the shape of the plot tells us if we have a high proportion of small aggregates, if it goes up more slowly, we have a higher proportion of bigger aggregates. And hopefully you can see by eye that the Alzheimer's disease CSF has a higher proportion of bigger aggregates because it goes up more slowly. And these aggregates, I should point out, range in size from 20 nanometers all the way up to 150 nanometers. So there's actually surprisingly large aggregates present in the CSF. This is an atomic force microscopy image of the same sample. This is the control, this is the MCI, this is the Alzheimer's disease. So if you look at the highest resolution images, the brightness just showed you how much the aggregates come up above the surface. For the control, you see a large number of small spots only coming up a small amount above the surface. If you look at the Alzheimer's disease patients, you can see the aggregates are much bigger and taller. They're brighter in the plot. And the MCI is sort of in between with some larger aggregates that are also brighter. So if you look at the height, this is the MCI, this is the Alzheimer's disease. So most of them about 0.5 nanometer, but the Alzheimer's disease also contains these aggregates that come up about 2.5 nanometers above the mica. If you look at the length, the control are very small, but the MCI are longer, and the Alzheimer's disease are even longer. So there isn't a crystal structure of toll-like receptor 4 binding to A beta aggregates, but there is a structure for toll-like receptor 3 binding to double-stranded RNA. So it binds hand over hand with the double-stranded RNA sitting inside the toll-like receptor 3. So we imagine that these protofilaments, which is what we're seeing in the Alzheimer's disease atomic force microscope images. So these are monomer binding end on end to monomer. We think that those would also be 2 nanometers in diameter, and we think that species would be very well bound by toll-like receptor 4, and that's why they're inflammatory. And then we think they're particularly inflammatory because they won't just bind one toll-like receptor 4, they're 100 nanometers in length, so they'll bind multiple toll-like receptors 4, and hence that they're multivalent and will trigger a much larger response. So this seems to give us a mechanism where to explain why these longer aggregates that we see in Alzheimer's disease patients are inflammatory. So what we think is happening is you start with monomer, you form oligomers, which seem to be good at permealizing the membrane, and then you get these protofilaments and protofibrils formed, which are much better at causing inflammation by toll-like receptor 4, and eventually you form fibrils. So as the disease progresses, what we imagine happening is you get a higher proportion of these longer aggregates, which become more toxic by an inflammatory mechanism. So the larger aggregates are formed as the disease progresses, and they're toxic by different mechanisms. And we think these protofibrils and protofilaments are inflammatory by toll-like receptor 4, suggesting toll-like receptor 4 might be a good receptor to target to cure the disease. What we find is if we add very low levels of aggregates, but over for long lengths of time, we can see this increase in the amount of TNF-alpha that's produced. This is just adding 10 picomolar of aggregates. If we knock out the toll-like receptor 4, we get no response. So this is for a cell line, and this is for astrocytes. So these low doses over extended times seem to give a sensitized increasing over time response to the aggregates at physiological concentrations. And what we found is if we do an experiment in a brain slice where you stimulate using electrodes, there's this phenomenon called long-term potentiation. So what happens is there's effectively a cellular memory response, and you get an enhanced signal. So the black spots are the control, and then when you excite the slice with particular pulses, you get this increased response. If you add A beta, what happens is it blocks this response. So you get these triangles leading to no response. But you can recover this response if you add A beta, but add an antagonist to toll-like receptor 4, suggesting that it's the A beta triggering an inflammatory response that's giving rise to the long-term potentiation deficit, and that can be rescued by blocking toll-like receptor 4 with an antagonist. So that suggests the inflammatory response induced by these aggregates is giving rise to the memory loss that seems to occur in Alzheimer's disease. Interestingly, we found increased amount of TNF-alpha production in the hippocampus compared to the cortex of these rat brains. So I'm going to switch now to looking at the aggregates that are present in cerebral spinal fluid and serum. So what we found both in serum and CSF, these are Parkinson's disease patients, there was no difference between the number of aggregates in the serum or the CSF. We just saw a higher number in the serum. But this is just examples of the images of these aggregates. So there's a range of sizes, some small, some slightly bigger. If we do these cumulative frequency plots, hopefully you can see that there is a difference. You can see that the serum of the PD patient goes up more slowly, showing us that there's a higher proportion of bigger aggregates, and that there's not a very big difference in the CSF. So this is a plot of the difference. You can see there's a clear difference in the serum and a smaller difference in the CSF. So in both cases, there's a higher proportion of bigger aggregates in the CSF and serum. If we do the AFM imaging, again, we see the same thing. We see these aggregates present, quite large aggregates. There's a clear difference between the Parkinson's disease patients and healthy controls, both in serum and CSF. So what we did is do our aptamer imaging of all the aggregates, which will be imaging both the A beta and synuclein aggregates, but we immunodepleted the alpha-synuclein. So all we're imaging is the A beta aggregates. We immunodepleted the A beta, so all we're imaging is the alpha-synuclein aggregates. And we did our aptamer imaging to detect what was left. And then we can work out the proportion of aggregates that are alpha-synuclein and the proportion that are A beta. And we took the ratio of the alpha-synuclein to the A beta. And what we found was very interesting. The Parkinson's disease patients had a higher proportion of alpha-synuclein aggregates and all sat in this range here, while the healthy controls had a lower proportion of alpha-synuclein aggregates and all sat in this range here. So you can see that even at the level of individual patients, we can nicely distinguish between healthy controls and people who have Parkinson's disease. And these were people who had only just been diagnosed with Parkinson's disease a year or so ago. So this seems to have quite a lot of potential for early diagnosis of the disease, with the caveat it's only a small number of samples that we've analysed. So we're quite excited about exploiting this to see if we have an early diagnostic for the disease. So the last example I'm going to show you is work done on brain. So what we do is get post-mortem brain from our brain bank, chop it into small chunks, incubate it in artificial CSF, then spin it to remove the cellular debris, and take the supernatant. And what Dominic Walsh and colleagues showed in a paper is this was a way of extracting the soluble toxic aggregates from the sample in a very gentle way without homogenizing the sample and breaking up insoluble aggregates. So we've been using this method to extract soluble aggregates present in the sample. and then we're going to image them using our super resolution methods. So we took Braak stage 3 brain, which is early stage disease, and we took all these different brain regions that are color coded here, the entorhinal cortex and hippocampus, frontal cortex, and visual association cortex, which only gets infected much later on in the disease. When we took these samples and did our inflammatory assay, we found that all the samples caused inflammation to a greater or lesser extent. All samples were toxic in our toxicity assay, again with some variation across the regions. What we showed is that they're inflammatory via toll-like receptor 4, because if we take cells that we've knocked out toll-like receptor 4, we get no response. So we have aggregates, we have something inside the soaked brain that is causing inflammation by toll-like receptor 4. What we did was add an antibody to A beta, 6E10, and that blocked the inflammation. So that shows that the inflammation is indeed induced by the A beta aggregates present in these soaked brain samples. So we imaged the size distribution of all these aggregates. So these examples, we found the hippocampus, it has smaller aggregates than the visual association cortex in some of the samples. If you look at the size distribution, it's much clearer. The visual association cortex goes up much more slowly than the hippocampus. Across all these brain regions, there wasn't an obvious trend in the size of the aggregates or an obvious trend in the length distribution. Down here, some examples of the aggregates. You can see the hippocampal aggregates and the visual association cortex aggregates. Again, we did AFM on these samples. In the hippocampus, we saw these fibrillar aggregates, but we didn't see them in the visual association cortex. We tended to see these larger spherical aggregates. So this is just quantifying this. We have more spherical aggregates in the visual association cortex, more elongated aggregates in the hippocampus, and this is their length and height. So it looks like in the hippocampus, we're getting these protofilaments, protofibril aggregates that are more inflammatory, maybe explaining why you get memory loss and more disease in the hippocampus at Braak stage 3. So to summarize this part of the talk, inflammatory aggregates appear to be everywhere in the brain to a greater or less extent. Inflamed microglia are also present in these regions at Braak stage 3, with more in the hippocampus. So it appears to us that the same degenerative process is occurring all over the brain, but to some regions at an earlier stage in the disease and some stages at a later stage of the disease. So rather than the disease spreading through the brain, there's just a gradient. The whole brain is undergoing changes driven, we think, by inflammation, but some regions have been inflamed for longer and having more inflammation, and that's what's driving the disease. So the last part of this talk, so one clear issue we have is that we can't distinguish between A beta and synuclein aggregates. So to address this, we use this so-called single molecule pulldown assay, which we adapted. So the idea is we have a passivated surface, it's normally a PEG surface, and we have streptavidin on this surface to which we attach an antibody labeled with biotin. So this might be an A beta antibody. It will capture the aggregate, and then we have an imaging antibody that also binds to the aggregate, and this will have a DNA strand, and we can do DNA paint to make it super resolve it. And so we can capture aggregates of A, we can capture aggregates of B, or we can capture mixed aggregates of A and B by capturing with A and then imaging with an antibody to A and an antibody to B. And this might be, for example, A beta aggregates that are also bound by ApoE. So we've actually demonstrated that this will work. So we can ask which of all the aggregates are ApoE associated. So just to show you how this works, this is, we attached 6E10 to the surface, captured A beta aggregates that we detected with another 6E10. This is the buffer, we see very little, and this is the aggregates. So we can see the individual aggregates on the surface, and we see a large difference in the number of aggregates. If we have the antibody that binds, that's the same for capture and detection. It shouldn't bind to monomer because there's only monomer will only have one epitope, but it should enable us to detect dimers or larger aggregates. This is another example, super resolving alpha-synuclein aggregates. So this is the typical way we build up the images. We get this blinking, so we can super resolve each DNA that binds to the antibody. And then we can build up a super resolution image of the aggregates. So we're now using this method so we can fingerprint the A beta aggregates, tau aggregates, and alpha-synuclein aggregates present in the sample. We can measure their size, and we can measure their number, and we can measure their shape, whether they're fibrillar or spherical. So we really just wanted to summarize. We can do imaging aggregates from humans, so we can use this to study the real disease, not models. These are generic methods that are applicable to other proteins. We're finding out how these aggregates initially damage neuronal cells. We think inflammation plays a key role, and that's via toll-like receptor 4. The method has potential for early diagnosis and also the potential to monitor the effectiveness of therapies. And we hope it will provide the basis for rational design of new therapies. And I'd just like to acknowledge all the people involved in the work. Dimitri Sidoris did all the work on the soap brain. Suman Day did the work on the CSF, or led in that work. And Eleni Dimu, who's in the DOI, did all the nice imaging of the tau aggregates. And there's a range of collaborators in Cambridge and beyond who've really assisted with this work. And I'll happily answer any questions that you have.
There we go. Thank you so much for a really, really fascinating talk and a really lovely mixture of technical versus also including then how it's used to answer the biological questions. So really, really interesting. Thank you. So if anybody has questions, feel free either to pop them in the chat, or if you'd like to unmute yourself or raise a hand, and we have a little bit of time for some questions. Perhaps in the meantime, I can kick off with the first one. So the toll-like receptor 4 seems to be a really dramatic response when you block that. Is this something that could be explored as a therapeutic option in vivo? Is there any sort of mouse studies that are being done on this, or is it something feasible in humans?
Yes, so we have a drug development project around toll-like receptor 4. There is a problem that toll-like receptor 4 in mice doesn't have the same sequence as in humans.
Right, okay.
Make some of the development difficult, plus questions around, what is a good model of the disease. But we've done two things. We've tested over 1,000 small molecules, and we found some that were low nanomolar binders, but unfortunately, they all got destroyed in the liver, so the pharmacokinetics wasn't very good. But conversely, we've also got a whole bunch of antibodies to toll-like receptor 4, which again have very high affinity, look very promising. So we're continuing to explore that route. So we think we might have some very high affinity binding antibodies that could bind to toll-like receptor 4 and switch off inflammation. And those would work very effectively, blocking any inflammation that's in the periphery. But we also think, because of what's been shown with anti-A beta aggregate antibodies, that they should be able to cross the blood-brain barrier if it's got high affinity and bind toll-like receptor 4 in the brain. So we're at the early stage. The funding we've got is essentially to get us to a phase 1 level, and then we can go from there. But I would say it's looking quite encouraging in terms of having a reagent that might be effective, and then we really need to test the hypothesis that it is toll-like receptor 4 that's playing a critical role in the disease there's quite a lot of controversy whether the inflammatory response is protective initially in the disease. But certainly people who've got symptoms of Alzheimer's, one would think the inflammatory response it's not protective, it's driving the disease. We imagine inflammation, and we've got data that shows that if you take cells and add TNF-alpha, that leads to increased production of aggregates. Those aggregates would lead to increased inflammation, increased production of pro-inflammatory cytokines, and then you have this cycle. And if you look at in the PET scans, the tau PET and the inflamed microglial PET spreads through the brain at the same time, suggesting it's a self-driving cycle. So we think it holds a lot of promise, but we've got quite a lot more to do.
Right. That's really interesting. And in terms of TLR4 physiologically, is there any role that you'd be concerned about blocking sort of outside of the aggregate story?
I think the argument is, it's our first line of defense, and we use it a lot when we don't have a adaptive immune response. But in older people, then you have an adaptive immune response, and blocking your innate immune response is not going to have much effect.
Yeah, right. And there's a comment that we have in the chat. This is from Marco, and he said, I'm sure you've already thought of it, but maybe a humanized mouse would help. And great work, by the way. Congratulations to you and the whole team.
Well, thank you. Well, I think it's a big discussion about, you can have a mouse that has humanized TLR4, but then we would need to cross it with an Alzheimer's disease mouse. So it becomes quite a lot of work, I think. But I mean, all these things are possible. I mean, the main issue with the mouse models is they're not real models of the disease, because you're overproducing A beta. You've turned A beta up to some high level, or tau, and you're asking, how does the mouse respond? And can you block the effects of this overproduction of A beta and tau? In reality, in humans, there isn't an overproduction that drives the disease. There's a normal level, and for some reason, something goes wrong, and you have accumulation of aggregates. So in my opinion, you're not really modeling the key question is why do you start to get aggregation? What leads to aggregation? And that isn't, we think it's not an overproduction, actually. We think the production is the same. It's probably a failure to remove the aggregates effectively, which leads to them becoming larger and more toxic. That's what we think happens. And so a lot of these models don't recapitulate that at all. So they're good asking questions about, if I overproduce A beta, can I block it with antibodies? But they're not a great model of the disease. So in our picture, and I haven't really shown you the data, but what we find is, it's not like you have no aggregates and then you develop Alzheimer's disease and suddenly you produce aggregates. What we find is normal cells produce aggregates all the time, but they remove them as fast as they're formed, so there's no accumulation of the aggregates. If you stress the cell, either stress the cell, well, essentially, if you stress the cell with some inflammatory stress or some cellular stress, what happens is the cell is not as effective at removing the aggregates, and they start to accumulate, and you start to see bigger aggregates, and you start to have cell death that leads to release of aggregates. And so you get the disease because this imbalance occurs. If you're unfortunate enough to have a mutation that means that you have a more aggregate-prone protein, then you need less stress to reach this tipping point, and you'll get the disease earlier. So you have to wait till 50 or 60 because the cells aren't exposed to sufficient stress to get to this tipping point. But then you get this positive cycle of aggregation and inflammation we think, driving the disease at that point.
That makes, yeah, that's very interesting. And I suppose in terms of that as well, so in Parkinson's, you showed obviously the larger aggregates were more, or the difference in the aggregates was more profound in the serum than in the CSF. That's correct, right? Based on
yes.
Yeah.
I mean, that's a surprise.
Yeah.
I think there's quite a lot of evidence. There's a recent Nature paper that the disease might occur in the periphery, you know, either in the gut or elsewhere in the body. You get essentially an autoimmune response to synuclein aggregates. So you get inflammation, you'll get T cells that can cross the blood-brain barrier, which are looking for synuclein aggregates and respond to that. So there's a picture, certainly for Parkinson's, that the whole process might not start in the brain, but might start in the periphery or the gut. And that, however, our data would support that. It's very surprising. It does suggest that there's synuclein aggregation occurring outside the brain. We actually saw 10-fold more aggregates in the blood than the CSF. So if it was all in the brain, one would imagine it gets diluted probably 10-fold in the blood, and then it gets removed and you'll see less aggregates in the serum. But we see more, which suggests there are aggregates coming from other parts of the body that are producing synuclein aggregates. Of course, there's an awful lot of synuclein in the red blood cells, but it does look like somewhere in the body, maybe the red blood cells, maybe somewhere else, you're producing aggregates. And this Nature paper would suggest that these are recognized by T cells, and they then recognize synuclein aggregates and then can get into the brain. So the whole thing might not be happening starting in the brain, which might be good news, but it might be much easier to intervene if it's not happening in the brain.
Yeah, absolutely. And these were early stage patients, if I recall. Have you had a chance to look also at much later stage patients to see if that balance is the same?
No, we haven't. I mean, I think we want to go earlier. So it's not a great benefit to diagnose people. So the doctors can quite quickly diagnose people with Parkinson's disease. I mean, they basically tell me you have to ask them to draw 2 interlocking pentagons and name 20 animals in a minute. And if you can't do that, you've got Parkinson's disease, as far as I'm concerned. So it's not particularly difficult to diagnose. I think the real question is, can we diagnose it earlier? So we would be interested in getting that people have these disrupted sleep patterns, and we can get blood from those people. And a lot of these will go on to get Parkinson. And if our marker is up in these people who haven't got the disease but are going to get the disease, I think that would actually have the opportunity for early diagnosis. Of course, there are questions around the fact there's no treatment, even if you get diagnosed. But I think the argument would be that if you got diagnosed earlier, treatments might be more effective before you develop the fully fledged disease. So there might be some opportunities if you could detect the disease 5 years before you actually got it. Some of the treatments that we've got might be much more effective at that stage than when you've got the fully fledged disease.
Yeah, yeah, that's really interesting. So do we have any other questions? Feel free to raise the hand or unmute yourselves if you have any questions at all. Just have a quick check in the chat. Everyone's been quiet today, it seems. No. Okay, well, in that case then, thank you so much for joining us. It's been really interesting to see the work and the techniques and what you can then gain from that. So thank you very, very much. And you're obviously welcome to join any of our future TMS meetings as well, if you're interested in any of the future meetings. But for now, we'll say a huge thank you And good luck with the next stage of the research.
Thank you very much. I really enjoyed giving the talk, so thank you.
Thanks so much.
Goodbye.
Next week.
Bye.